So, good morning. Today's topic for discussion is cytokine and effector functions. So, what are cytokines? They are small soluble molecules. So, they are protein secreted by the cells to mediate and regulate immunity, inflammation, and hematopoiesis. So they act through a specific receptor, initiating signaling cascades, and they also influence proliferation, differentiation, and effector functions. So understanding the cytokines and their interactions is crucial for the comprehending and immune responses, and also to understand how developing therapeutic intervention works. So coming to, there are several kind of receptors, different class of receptors in cytokines. So they are classified into several families based on their structural similarity and receptor usage. So there is hematopoietin family. It includes interleukin, example IL-1 and IL-2 and IL-4, and growth factors such as erythropoietin. Then there is interferon family. It comprises of type 1 INF alpha beta and type 2 INF gamma interferons, which is crucial for antiviral response. Then there is TNF, which is tumor necrosis factor family. It encompasses of TNF alpha and TNF beta, and it is and related ligands involved in the inflammation and apoptosis. Then comes interleukin 1 IL-1 family. So it includes interleukin 1 alpha, 1 beta, and 1A. So they are key mediators for inflammatory response. Then there is transforming growth factor. It is TGF beta family. So this regulates immune suppression and tissue repair. Then there is chemokines. It is a large family of chemokines guiding cell migration. They are divided into CC, CH3C, and XC chemokines. So there are different types of chemokines based on the cysteine motifs. So how does the signaling occurs? There are basically three pathways that we will see today. The most common of them. So cytokine binding receptors undergo conformational changes, activating intracellular signaling cascades. So these pathway ensures precise control of immune response with dysregulation, leading to pathological conditions. There is JAK-STAT pathway. So Janus kinase is the phosphorylate signaling transductase and activators of transcription STAT. So which dimerizes the dimerizes and translocate the nucleus, modulate gene expression. So in this we can see the first, there is recombinant cytokines which attaches to the receptors, and then it produces a small molecular inhibitor that will affect the DNA function. Then there is MAPK pathway. So this is a mitogen activated protein kinase, MAPK, and they rely on signals affecting the proliferation and differentiation. Then there is NF-kappa B pathway. It is a nuclear factor kappa light chain enhancer activated B cells. So this mechanism controls the gene involved in the inflammation and the survival of the cells. Coming to effector T cells and cytotoxicity. So effector T cells primarily CD8 plus cells, cytotoxic lymphocytes, the CTLs, cytotoxic T lymphocytes. So these eliminate infected or malignant cells. That is their role. And these mechanisms ensure the targeted elimination of compromised cells while sparing healthy tissues. So they only target the affected cells that have turned malignant or got infected by other pathogens. So the activation, the naive CD8 plus cells as seen here. So they recognize the antigen peptides presented by the MHC class molecules receiving the post-immunity signals for activation. Then the cytotoxic mechanism, they have basically two cytotoxic mechanisms, perforin and granizymes. So perforin forms pores in the target cells and cell membranes, allowing granizymes to enter and induce apoptosis. Also, there is Fas-Facile interaction. So engagement of facile ligands on the CTLs with the F-Fas receptors on the targets, it will trigger apoptotic pathways. This is the image that is shown here. That shows the cytotoxic mechanism. It will the molecules will go inside and it will trigger the. This is the image of perforin. The molecules will go inside the granizymes and it will trigger apoptosis. Then there is natural killer cells and antibody dependent cellular cytotoxicity. So what are natural killer cells? They are innate lymphoid cells capable of destroying and or transform cells without the prior sensation. So NK cells basically are the killer cells of body. They will kill all the cells that are marked. And NK cells also secrete cytokines like INF gamma, modulating adaptive immune response. So the recognition, this detects cells with the reduced MHC class 1 expression or stress-induced ligands, like I said before. And the mechanism of cytotoxicity is the perforin and granizymes is same. It will similar to CTLs. The NK cells induce apoptosis in the target cell. Also, there is another mechanism, that is cell death receptor pathways. So, on engagement of TRAIL or FASL with the receptors on the targets will initiate apoptosis. The mechanism of target killing cell. So the immune cells employed various strategies to eliminate targets. So these mechanisms will ensure the effective clearance of infected or malignant cells while maintaining the immune response, immune regulation. There is granule exocytosis. So that is the release of perforin and granizymes to induce apoptosis. There is cytokine mediated killing, that is secretion of TNF alpha or INF gamma, which can induce apoptosis or inhibit proliferation. Then there is death receptor engagement. So activation of Fas or TRAL receptors triggers apoptotic signaling. There are some of the few recent researches that have happened in this topic. So there is cytokine adapters. So that is development of molecules converting immunosuppressive cytokines, for example IL-10 or TGF-beta, into immunostimulatory agents that can enhance the T cell responses. Also, there is interleukin 21 in natural NK cells function. So studies highlight that IL-21's role in NK cell maturation and cytotoxicity, suggesting a therapeutic potential to enhance the innate immunity. Autophagy and T cells, autophagy in T cells. So the researchers have revealed that the antigen receptors engagement represses autophagy in the T cells, influencing differentiation and function. Also, the natural killer cells in cancer immunotherapy. So advancements in understanding the NK cell roles in tumor immunity have led to the novel therapeutic strategies targeting the NK cell pathways. So that's for the topic today. Thank you.